Hey, Eric, you there?
Yep, I'm here.
Joel, you still there
Hey.
also?
Yep. Yep. How you doing?
Awesome. Good.
Hey everybody, Mike here. We've got a special show for you here today. That was the sound of Matt and Jody calling two people. One of them is none other than our very own Dr. Battaglioli, also known as the Wild Health Director of Microbiome, a name that we gave him mostly because Dr. Poopsack was already taken by a gastroenterologist in West Texas. And the other is our guest for this podcast, Dr. Joel Dudley. In today's podcast, you're going to learn all about something Onegevity is pioneering called multi-genomics. You're also going to learn a little bit about their ideas for N-of-1 trials with the microbiome, and maybe a few more practical things like should you take probiotics, should you take prebiotics, or both, or none. Dr. Dudley is co-founder of Onegevity, a company you may have heard of. It's pretty much at the leading edge of microbiome testing. He's currently associate professor of genetics and genomic sciences and founding director of the Institute for Next Generation Healthcare at the Icahn School of Medicine at Mount Sinai. He also serves as executive vice president for precision health of the Mount Sinai Health System, and his work is focused in the nexus of omics, digital health, artificial intelligence, scientific wellness, healthcare delivery. His work has been featured in New York Times, Wall Street Journal, Scientific America, MIT Technology Review, CNBC, lots of other popular media outlets. He was named in 2014 as one of the 100 most creative people in business by Fast Company magazine. He's co-author of a book exploring personal genomics from the Oxford University Press. Dr. Dudley received his BS in microbiology from Arizona State University, his MS and PhD in biomedical informatics from Stanford University School of Medicine. So I guess what I'm trying to say is this guy knows a thing or two about this stuff. So Matt, how about you just tear into him with some questions like you don't have any manners?
I've got a problem. I saw a patient yesterday, and what we do at Wild
Hmm.
Health when we see patients is we take a lot of data. We do their genomics. We run it through a couple different programs with some machine learning and AI, and then just look at a bunch of SNPs in general. And then we correlate
Hmm.
that with their lab data, their microbiome data, looking at all their hormones, kind of some advanced hormone testing. And then also we take into account their lifestyle, their social situation, if they have a mindfulness practice, a lot of data and information. Um, and then I try to come up with kind of a precision plan for them, but it's a lot of work and a lot of time.
Hmm.
And
Hmm.
I think maybe you can help. Am I right?
Yeah. Yeah, I hope so.
Awesome.
Yeah.
Hey there, I'm Mike.
And I'm Matt.
And I'm Jody. The Wild Health Podcast is our quest into human performance, longevity, and personalized medicine. Together, along with experts in these fields, we'll learn how to live longer, better, and become smarter and stronger. We'll do the deep dive into the research and hard work of putting it all together, and you reap the benefits.
We've been looking at it and kind of reading and listening to podcasts about Onegevity, this new company that's doing microbiome based personalized diagnostics and optimization services. And we're really excited about their potential to maybe replace our human brain a little bit and do a little bit of the legwork up front so we can spend more time just talking to patients. So, Dr. Dudley, could you start off just kind of telling us what Onegevity does and how it's different from other microbiome based programs?
Yeah, sure. So we co-founded Longevity with Chris Mason, who's a professor at Cornell, and it's really a company that has been brewing in our minds for a long time. For one reason, based on the research that we've done for the past decade, he and I are both sort of been in the integrative genomics space, right? So never looking at just one dimension of data at a time, always trying to build these sort of integrative models that combine multiple modalities of molecular data to really give us a bigger picture of what's going on, say, in a particular disease that we're trying to study. And then also, based on the fact that I myself have Crohn's disease, and I'm an early adopter, right? I've tried everything that's out there. I went to school with graduate school with some of the co-founders of 23andMe. I knew the Ubiome folks, for example, before they got started. So I've always been an early adopter of this stuff, and I love it. But really, I felt that what was out on the market was just not adequate. I felt that a lot of the companies that were out there offering these types of tests and were sort of recreational health information companies, I call them. So you did your test and you got all kinds of cool stuff, information back, but then there's the now what part of it, right? Which left me and others to sort of figure out what comes after that, right? So the idea behind Onegevity is we're building a company that's a lot less like, and there's nothing wrong with those companies that are out there that I mentioned, by the way. They're doing, I think, fairly okay, but we're taking a different angle, which is, and some people might be familiar with a company called Stitch Fix, which is actually a clothing company. But what Stitch Fix does is you kind of put in your preferences for the clothing that you like, and then they have a whole data science team behind it, and it sort of feels like you have a personal shopper, because they send you clothes, and then you keep what you like, and then you send back what you don't like, and on the back end is actually data science and algorithms constantly learning your preferences and what you like and don't like, and sending you new clothes and becoming your ultimate sort of personal shopper. So anyway, that's actually more the analogy behind what we're building at Onegevity. So we're less like these other testing companies, and we're really trying to build a company that's more like Stitch Fix, except we'll test you through multiple modalities, and then we'll actually deliver the solution to the problems that we uncover in a totally personalized way through the testing.
Yeah, no, that's really exciting, and that's what excites us, because we've kind of heard you say that and read that. You mentioned the other kind of testing companies. We actually recorded a podcast with not to name names, but the biggest other microbiome testing company, and we have actually not released the podcast because we were not happy with the chief science officer's answers to the questions and the science they were using, and we just didn't feel like it was probably useful enough to our patients. But we're excited about what you're doing. We think it could be the solution. That's why we want to dig into that. I also have Dr. Eric Battaglioli on the line. He is our director of microbiome for Wild Health, and he can ask much more intelligent questions about this than I can. So, Eric, I think you potentially want to talk about multi-omics in general. Is that right?
Yeah, that's right. So I think getting right to it, that the integration of multi-omics platforms is going to be one of the bigger goals that's going to move the needle forward and trying to bring all of these different facets of information together into a coherent outcome for an individual person. So I'm curious about the multi-omics platforms that Onegevity is using, and how you're incorporating genomics, serum, untargeted and targeted metabolomics, and microbiome information to improve the outputs of the analysis and affect individualized care.
Yeah, sure. And that's really been the core of my work and Chris's work on the research side. And the challenge there is, we've applied this a lot on population scale data, and then now what we're doing at Onegevity is how do you apply that to the individual. So there's a couple of different approaches on how you can integrate this type of data, and the approach that I've used a lot in my academic work is various types of network modeling, where basically, instead of just looking at every dimension of data on its own, and then after the fact trying to piece together a story, after you sort of analyze each individual data type, you do, say, a type of integrative network modeling to various forms of that, Bayesian network modeling, other types of network modeling, where you can sort of build a totally integrated model that tries to understand how the information flows between all these different layers. An example of this was a study we had published recently out of my research lab on Alzheimer's disease, and we sort of combined proteomics, DNA, RNA, clinical data, and a bunch of other dimensions on a large cohort of Alzheimer's patients, and that was a discovery study, but we revealed in a totally unexpected way that herpes viruses were sort of participating in these pathways that in the Alzheimer's brain. So the power of that integrated approach is really manifold, but two important ones are really understanding how information is flowing between these different layers of information, and then figuring out what or learning things that it can be totally unexpected, that are sort of hiding in the data. And then, of course, the big challenge within all of this, which is sorting out, correlation from causation in big data sets, which is actually a huge problem in general. But there are techniques you can use in these sort of more directed network models. You can use from the DNA expression quantitative trait locus sort of information, which is sort of the correlations between SNPs and gene expression patterns, and sort of apply some statistics that help you sort of sort out the correlation from the causation. But at least before diving too deep into that, it's pretty complex, but sort of the high level approaches.
Yeah, I think that's awesome, and really a direction that these types of analyses need to go to be able to sort out the difference between the signal to noise ratio in those data sets. So you mentioned as sort of like a testing platform, looking at large population scale data sets as a way to establish a network reference range. How does that play into, and that's a question we often have for individuals and companies in this space, is how do you define normal when there's so much complexity to this system, and how do you create or establish a reference range to train a neural network or train a network model to be able to apply it to a broader audience?
Yeah, that's a great question. And one thing I've been saying too recently is people don't realize we're sort of starting over in microbiome space. So what I mean by that is, so our product, the Gut Bio product, is a shotgun metagenome product, and a lot of the data sets that are out in the public domain, and also products offered by other companies, are 16S microbiome sequencing, and it's pretty hard for, I think, most people to understand the difference between those. But the analogy I've been using lately is 16S is like a black and white TV from the 1950s, and shotgun metagenomics is like a 4K Ultra HD TV. That's the difference in resolution you get. But it generates a lot more sequencing data, and not until very recently has it become more economical. But what that means, though, is now we need to sort of wait for the large shotgun metagenome reference populations to build up and be published, and they're starting to happen. But we benefit from, in that regard, from the work of Chris Mason, who's one of the co-founders of Onegevity. He's kind of a crazy guy, and he's been basically microbiome sequencing anything that he can touch for the most part. He has a project to, in the past, where I always joke that he sequenced New York City's gut microbiome, because he sequenced the entire New York City subway system, and which was revealed all kinds of interesting things. And in fact, when they did that study several years ago to sequence the New York City subway system, they actually found, I think, 48% of the species they found were novel, or some crazy high number like that. Wow. Because they're using shotgun metagenomes. And now he's actually got a project called Metasub, where they're trying to sequence every subway system in the world currently. So that, through his work and some consortia and things he's involved in, we're fortunate enough to start to get access to large reference data sets of shotgun metagenome data to help us sort of tune, and figure out. You're exactly right that we don't know what normal is in the environment around us, and then also what is normal sort of in our bodies.
I do want to talk specifically about the microbiome testing you are doing, but right before we really get into that, do you this multi-omics approach that you're kind of talking to and alluding to? It's kind of, there's a roadmap that you all have for that. Is it too preliminary to kind of tell us when you'll be able to apply those multi-omics and not just do the microbiome testing? Because that's really exciting to me, but you're not doing it quite yet. Yeah. Do we know when?
Yeah. Oh, yeah. So, and it's part of a roadmap of rolling out both the testing and then, the software. So the genetics component will come out within the next few months. We announced a partnership with this company, GenCode, which is based in New York, and it allows us to do very low cost whole genome sequencing through the sort of the software platform they've got. So I think, I'm not sure what the final price is, sub $100 full genome, basically. So the genetics component is coming soon, and then there's there's actually quite a few known, and then also new types of I talked about expression quantitative trait loci, which are sort of correlations between SNPs and expression, but there's also MQTLs, or microbe, quantitative trait loci, where basically we can find sort of correlations between the host SNP markers and the microbiome species and things like that. So that's one of several ways you can integrate those data to really understand interactions between those things. And then obviously, I think the exciting component and a lot of this will be the blood testing, which we're going to roll out to this Drawbridge Health device, this painless self-administer blood draw device. That device is currently at the FDA, and we expect that will be resolved sometime this year. But we're waiting to hear back from the FDA, and then we can move forward, with offering that.
Yeah, that's really exciting. Right now, when I've got hundreds of pages sitting in front of me with someone's genomic data, and then also all their blood testing, and then also their hormone, and then also their microbiome, it can be overwhelming. So it's going to be great, I think, to have that in one place. Right now, with the microbiome, how do you use I've looked at some of your reports, but I want to hear you describe how do you use this information to make specific recommendations? Because that's always the complaint when I have patients come to me who they've had their microbiome tested with other companies, and they get this data and it tells them things, but doesn't really tell them what to do usually. Do you have kind of an example of how that would work, what kind of recommendation it may make to someone, and how you arrive at that recommendation?
It's a combination of, in terms of the underlying information, it's a combination of our own data within the company, and then also a lot of it is bootstrapped, as it is at most other, firms, is by synthesizing the knowledge that's out there in the world, right? So there's a lot of microbiome food associations, microbiome disease associations, and you could sort of, basically go through the literature and curate these things, and we've done that. So we sort of built a structured internal knowledge base of everything that's been published, and then, but as anyone who does an academic research, you really can't take the published research at face value. You got to do a lot of curation and use your own expertise to figure out what is likely to be true and what is likely to be less reliable in terms of what's published. And then there's challenges in terms of how the associations were described. Were they at the sort of the species level or just the genus level, or so there's a lot of things you have to think about. It's when you take the information that's already been published, and how you normalize that data and structure it and compare it. So that's a component of how we bring in some of the recommendations by synthesizing in our own sort of proprietary way, I guess, the published data. But we're also conducting our own internal studies. So we will hope to publish very soon in IBS a trial that we conducted where individuals were given selections, a customized selection of products based on, and in this study it was Thorne products. Thorne is is the first company we're working with that offers products that could potentially be used to deal with issues in the microbiome, although Thorne will not be the only company. It's products that we work with. We're free to and will be engaging other solutions.
Well, just to give you a specific example, so you're not kind of guessing what I'm thinking. I specifically was thinking about a patient of mine who sent me their Onegevity data, and a specific recommendation that came up was take a Thorne B6 supplement based on looking at their gut microbiome and saying that based on the species that are there, you're probably going to be deficient in some of the vitamin B6 complexes. Niacin, I think, was one of the specific ones. Do you all have studies specifically on those recommendations and whether they actually improve outcomes, or is it just a kind of, this makes sense, so you should do this?
Yeah, it's a mix of both. So some of those it makes sense. In that particular case, we're able to do, because we're doing metagenomics, we're able to infer, a bit more about the sort of biological activity or pathways, if you will, of the species, that are in the gut. Because metagenomics just provides so much more information. We can actually infer quite a bit more about the gut metabolism. In fact, one of the features we're working on, which I don't believe is rolled out yet in the product, is something we call virtual metabolome, where basically you can, and we have the science behind it, but basically you can infer a lot of the fecal metabolome just from the content you get in the raw reads of the shotgun metagenome profile. The way you do that is, we have reference databases where the shotgun metagenomics data that's been collected, and on the same samples they've done targeted metabolomics, and which it's really important to know what metabolites might be produced. But collecting metabolites or untargeted metabolomic data from fecal samples is a very logistically challenging process. So it's not, you need refrigeration and dry ice, and there's a lot of volatility in the sample. But what we're showing, for example, in our R&D pipeline, is you can actually, using deep learning algorithms, infer a lot of the metabolome from the shotgun metagenome read. So that's the B vitamin example is sort of a bacterial metabolite in a way that we would be inferring potentially from the data or by literature association. But we are conducting our own studies as well with the Thorne product. So as I mentioned, with around IBS, we just concluded a trial, and people had an intake questionnaire, but we also did the metagenome, and the number of product combinations that were possible was vast. I can't remember the exact number, but above 100 possible different product combinations, and we then customized the products based on their microbiome, and then did symptom scores and other sorts of measures around IBS outcomes, and it was extremely promising and positive response in that trial in terms of figuring out what worked and what didn't. And overall, it worked well, but it also helped to sharpen our algorithms even more in terms of the predictions. And we'll be publishing that study soon.
So you are looking at symptom resolution and measuring these outcomes specifically.
Oh, absolutely.
Yeah, that's great.
It's just, as fast as we can conduct these studies and keep building up the evidence base around these. It's because there are big holes in the literature, so you can't just rely on the literature alone.
Yeah, no, that's exciting. That's what we care about, whether the patient's getting better or not, not just getting their levels up. So that's incredible. Now, when drawbridge and the serum testing is brought in as well, would that go into the algorithm for the recommendation also? Would you be looking at those actual levels and not just the gut microbiome?
Yeah, exactly. Right. Yeah. So the beauty of being able to do the blood then is we can actually, so we can make a prediction about the metabolomics, say, for the gut, but obviously you can verify what is actually getting into the blood, for example, and actually obviously lots of other useful things. Lots of blood, but really being more direct about understanding those connections if you want to test for B vitamin deficiency in that particular case.
Now, if someone has kind of similar microbial community, say their lab testing looks similar from Drawbridge and these other platforms you're using, you mentioned a questionnaire. Are you also able to roll into this things like different lifestyles, genetic backgrounds, health status, their kind of dietary preferences, whether they're vegan from an ethical standpoint? Are you able to take that into account as well for when making recommendations?
Oh yeah, absolutely. And I think we already do to some degree, and that's something that we're always trying to improve. And it's interesting too, by the way, is something you can already see in the data is you can have people tell you if they're vegan or whatever. Yeah, it's actually pretty easy to guess from the microbiome what they are or not. I mean, it's hard to distinguish, of course, a meat eater who's also eating lots of plants, but it's pretty interesting when you start looking at the data. It's pretty easy to sort people out by diet, lots of lifestyle things and dietary preferences, and even by the way, medications like not over the counter prescription medications that they're taking. You can easily see in their microbiome whether they're taking them or not. It's pretty interesting. The sort of the richness of that data.
Yeah, that's exciting. It's one of the kind of challenges we have sometimes. You have someone with religious beliefs, for example, and they're not going to eat animal products, but at the same time, they may have a poor omega-3 to omega-6 ratio, and their genomics mean that they can't convert ALA into the active form of omega-3, and so it's difficult. And so if you were able to pull that into the algorithm, that would be incredible. It would save the practitioners a lot of time.
Yeah, we're working and talking to several companies that, as I mentioned, the whole point of what we're trying to do at Onegevity is to deliver the solution and make it easy for the consumer. And we are talking to various food companies that have the potential to customize and deliver the diet in this particular case of what meets all those needs. But you're right in that the degree of customization can be significant in some of these cases. So that's the challenge logistically.
Mm-hmm. Yeah, I'm hoping that you all mostly put me out of business, and so that I can just focus on a small part of taking care of patients and not all of that. Now, what if a person doesn't really have a problem per se? I mean, should they still seek to characterize their microbiome and alter their diet? What about those people who really don't have any problems? Is there much value in this testing for them?
I would say yes, because and the analogy is probably maybe someone who's otherwise healthy. Would you recommend that they go to the gym and exercise and do strength training? Right. This is probably an analogy here. Well, you don't have diabetes, or you're otherwise perfectly healthy, but why don't you hit the gym 3 days a week and just build up your muscles a little bit? Because we know in the future, as you start to age, that having more muscle mass and being more fit, and this is going to help build up a resilience that's going to protect you from disease in the future. And I think that analogy could be carried to the microbiome as well. So you're not experiencing any gut symptoms, maybe gas or bloating, but getting things sorted out, strengthening up the Akkermansia or other sorts of beneficial species that sort of boost the overall health of the microbiome. I think you could make a case that building that up when you're healthy could potentially save you down the road as age creeps in and other lifestyle factors might assault your health.
Yeah, no, that's great. I mean, in genomics, we frequently see a very healthy person that will find like an ApoE4 or something like that. They feel great. It gives us some guidance for the future. So it sounds like there's some similar things with the microbiome as well. Some good information there.
Mm-hmm. Mm-hmm.
So I have a question that's kind of an extension of that, which is, the average person goes in and says, I don't have a problem per se, but I'm looking to see what my microbiome's doing and try and push it in a more ideal direction. The assumption with that is there is an idealized microbiome, or there's a composition or a function that is best for the average person. And my experience in this case is it's been really, really hard to define that. So I'm curious if we're going to embark on the path of trying to push the average person's microbiome in a direction, how do we pick a direction, and what direction should we focus on?
Yeah, that's a great question. And I think for what we absolutely don't know yet, nobody could claim to know, in my opinion, is what is the ideal microbiome look like? I don't think anybody knows. I think we're still going to keep discovering new species in people's microbiomes as time goes on, just because we're just mapping it out, right? So there's a couple different ways you can potentially define that. One is there's a handful of species just for whatever reason seem to be always associated with doesn't mean they're causative, but those seem to be associated with reduced risk of disease, etc. But focusing on one species alone is just not generally a great approach, in my opinion. Right? It's an ecosystem in your gut that needs to be balanced, right? And I think this is part of the problem with probiotics, right? Because you tend to focus on just one species and hope that that's somehow a solution, but really it's a whole ecosystem you're trying to balance, and probably the composition of that ecosystem is more important than potentially any individual species, outside of having a dominant C. diff or something like that that's making your life miserable. But so we're still trying to figure that out, and as we're building up the data bases and also in some of the emerging data sets that are coming out of academia, there are efforts to sort of sequence or characterize sort of exceptionally healthy people from a gut health perspective, people that seem to have extreme resilience to disease, right? So I guess it would be an approach of saying, well, let's try to try to look as much as possible like someone who is has been extremely healthy from a gut health perspective. But then, of course, even then, that doesn't guarantee that that's the optimal profile, because you have to take into consideration, the background genetics of that population you're trying to mimic, and other environmental things and sorts of things. But I think in the short term, the approach will be, and I think for many people, because there's not many other options, is one, just using sort of population level comparisons, and if you will, sort of trying to move your microbiome into the GPS coordinates of other people that seem to be generally, exceptionally healthy in their gut. The second approach to that would be and this is something I'm working on the research side, which is we're developing software for conducting N of 1 trials, trials on a single individual, and which presents all kinds of statistical challenges when you're doing a trial on a single person. But we're working on some software that we hope to eventually license out. That the other approach would be, well, I'm just going to start experimenting on myself and try to optimize my gut microbiome through sort of a trial and error. So if someone wanted to be more aggressive about it, so that would be, I think, a second approach to that.
Yeah, and in personalized medicine, the N of 1 experiments is what we're all about. So that's exciting to hear you say that.
Yeah.
Since you mentioned kind of probiotics, I've got to ask the question about that. When I looked at the patient I mentioned earlier, their data, one of the recommendations was probiotics for them. And I think it was September last year, maybe in Cell, I don't know if you saw this paper, I'm sure you did, because you're in that space, talking about probiotics and the increased time to gut microbiome diversity when someone takes probiotics. It just gave me pause with probiotics in general. It made me rethink how quickly I reach for and think about probiotics, with it just being a few species and diversity being so important. I would love to get your thoughts on that in general. Did that study change at all what you think about probiotics, or how are you applying and when are you applying probiotics to a patient?
Yeah, so there was a study in Cell by this the Israeli group, great research group, by the way, who looked at probiotics, and to what degree do they take exogenous probiotics? Do they take up residence in the gut? And it's actually matching what they found matches a lot of what our internal data shows as well, which is or at least the belief that you can consume an exogenous probiotic, and that it's going to magically sort of sort of plant in your gut and just sort of sort of be like a silver bullet and make that change to your gut. That is, it turned out, looks like it's not the case. I always like to remind people that the microbiome is sort of like a garden that's already got totally planted. There's already weeds and flowers and plants it's already occupied them, and the bacteria that are occupying all those niches in your gut, they definitely do not want to give up that space. They're squatting and they're not moving. So when you eat stuff, right, it's difficult for exogenous species to just come in and just find root in your gut, because it's all occupied already. And and that's really, I think, what that study definitely showed is they have effects, but probably more transient for the most part. But it doesn't mean they're not valuable, right? So it's that the effects though are probably more transient, and it's probably more of a stimulation of there's many, you can think of many potential mechanisms that probiotics would still be useful, even though they're not really sort of planting themselves in your gut by stimulating other species, by stimulating your immune system in the right ways, etc. But what we find, and I think what the research is showing, and we found this actually quite strongly in our IBS trial, was that prebiotics seem to be a much more effective tool for sort of long-term or lasting modulation of the microbiome than probiotics, right? And that's selectively feeding what's already there versus trying to introduce something new from the outside.
Yeah, we've moved since that study, especially. I've moved more towards prebiotics and especially whole food sources of prebiotics and inulin and things like that. And I'm someone like you who has a much better understanding of it than I do. I'm glad to hear that you're kind of confirming that thought. One question that may sound kind of pointed, I don't want it to be, but I feel like I just have to ask it. In these recommendations, you kind of recommend Thorne products, which I think are great products, which I recommend. But there's obviously a little bit of of a conflict there partnering with Thorne. Is there any specific things you are doing to kind of mitigate that, or how do you think about that in general?
Yeah. So even though Thorne was one of our initial investors, they actually don't. We're not beholden to using their products exclusively. If they have a product and we want to use it, right, in a certain area, but they don't make everything. And there might be areas where another company's product has an extra ingredient or something we think would be beneficial. Let's say so. So I can guarantee that the business relationship between Onegevity and Thorne is set up in a way that Onegevity has a huge amount of autonomy, and actually the reason we really like working with Thorne products is we got to do a sort of full audit of their whole manufacturing process. I've been to their plant which is in Charleston, South Carolina, and we see how they make things, and it is just unbelievable. And obviously take it for the grain of salt from me, but their manufacturing standards, it's pharmaceutical grade. It's just unbelievable quality, and to us, we wanted to feel really good about what we were recommending, and we've seen it from when it comes into the raw materials and the detailed testing they do on purity, and that's just why we're excited to work with them initially. But if you think about where we're going with Onegevity, is we're going to empower individuals to really verify for themselves whether products are working or not. And this is actually what Thorne was excited about. And people might not think that's weird that a supplement company would want to empower individuals with the means to verify if the products are working or not, right? And when I met the CEO of Thorne, they basically said, I can make anything. I want to make stuff that works, right? And so there, which was, so that's basically a long game that they're playing, right? So the long game is that if we empower patients and consumers to verify the effects of our products, then in the long run, that's a better business strategy. So that's why we chose to work with them. There are other, people we've talked to that we're not so excited about giving people the ability to verify whether the products work or not. And Thorne was extremely open to that. So
no, that's great.
I'll leave it at that.
Well, it's great to hear that. I'm always very skeptical about pharmaceutical companies and supplement companies in general, but it seems like Thorne really is doing it right. So I'm glad to hear you say that.
Well, I was going to say the other thing with Onegevity and the relationship to Thorne and the ability to deliver products, and because we're seeking to make eventually our money from the delivery of products, that actually frees us up in a way to be more open with the data. So there's a lot of companies out there, the testing companies, that are their key value proposition is building big data sets, and really keeping those proprietary for the most part, and then that's their long-term monetization strategy, right? Is to partner, let's say, with a pharmaceutical company or something like that, and keep this siloed, proprietary data set. But I think one of the things that Chris and I are most excited about being academic researchers too is that because the value proposition of the company comes in really delivering products that solve problems for people, that sort of being greedy with our data is not required. So we hope to actually start to open up, with appropriate, consent from customers and things, our data set to help other scientists who are doing work in this area as well. So I think it's one of the areas we're most excited about, is that our business model allows for more openness of the insights and the data we're collecting.
Yeah, that is exciting. But I think a lot of people would be a little bit scared by that, just thinking about their data getting out. So how will you protect and make sure that their specific data is not going to trace back to them? And
oh yeah, no. So we're being, there's a very sophisticated, if we go in that direction, there's a sophisticated consent process, because we fundamentally, first of all, believe that the customer, owns all their data. That's their right, is to get access to their data and do whatever they want with it. And we're currently working on incentive schemes such that if the data is used in aggregate, so say you've opted in to your data for research, and then something comes of it, we do partner with somebody else, and then there's some value extracted from that data in aggregate. How do we share that value back with the customer, right? So sort of recognizing the role that customers have played, and there are lots of companies making money on data, on other people's data, and the benefits of that never trickle back to the person who donated that data. So the reason we haven't engaged in that yet is because we're actually trying to come up with very clear and transparent profit sharing schemes, essentially, that would then share the rewards of sort of these types of data analysis back with the customer. So that's what we hope to figure out here.
Yeah, that's really interesting. So just to make sure I understand, so the clients or patients themselves may actually financially benefit from a future relationship that you all may. Wow, that's really cool. So
yeah, exactly. Yeah.
So they're kind of investing not just in their health by getting this test, but investing a little bit in you all, and there may be a little bit of a return there, it sounds like. Is that going a little too far?
No, there's a lot of details to work out there on how you put that into practice, and there's even financial regulations around how you do something like that. So that's what we're sorting through. But we firmly believe that if we do make money on insights from the aggregate data, and if your data was part of that insight, that you've obviously consented to up front, we wouldn't look at it without that consent. Then we want to be able to share that the rewards of that back with you in a way that's equitable. So we're just working out the various models on how you might do that, but that's something we hope to roll out soon.
Yeah, that's incredibly cool. This question may kind of show some of my ignorance of the topic, but I want to ask it just to make sure I'm thinking about this right and get your thoughts on it. When you think about the gut microbiome and there being kind of tens of thousands of different microbes present, my understanding of the kind of shotgun metagenomics, it gives you improved taxonomic resolution but limited depth, meaning it may be kind of difficult to see the lower abundance members in the sample because you're kind of dominated by a few organisms. Do you think there's any information that we're kind of missing at the fringes, or do you feel like it's probably just not that important?
No, I mean, and this is actually a lot of the smarts that goes into our platform that comes again from Chris Mason's lab, because he has been really on the cutting edge of metagenome sequencing, all the way down to he can have a conversation with you all the way down to the biochemistry of what's happening on the sequencer and everything. But he's dealt with so much shotgun metagenomics data that they've had to really deal with these real issues that you just mentioned, which is the reads can get biased towards more abundant species. How do you detect the less frequent species? And fortunately for us, is there are computational ways to sort of deal with these issues and improve the fidelity of the sequence. So the same exact next-gen sequencing data set in our pipeline versus other people's pipeline is really benefiting from Chris's experience in this area, and how you can clean up that signal, how you can use machine learning and deep learning based approaches to boost in a high fidelity way the low frequency species and things like that. So that's a lot of the secret sauce that goes into our platform from Chris.
Yeah, that's incredible. You talk about the pipeline, and I'm always nervous about making a simple change from just a microbiome data or anything like that. But it looks like you all are taking into account a lot of these dynamic changes that may occur from a recommendation, and you're going to be testing for the results of those to really look at kind of host level effects instead of just the microbiome. So it's really exciting. And you kind of you confessed to us when we started that it's 7 a.m. your time. You're on the West Coast, and you had a good time last night. So we don't want to take too much more of your time. We know your time is valuable, and we really appreciate you coming on, because this has been really informative for me.
Yeah, well, we're grateful for your interest in what we're doing, and I hope we can keep living up to your expectations of what's needed and really help the patients you're trying to help.
Well, and as soon as some of these other things come online, the drawbridge testing, the genomics and things, we'd love to talk to you some more about how to use those to our patients' benefit. So we'll definitely be following up with you. Thanks so much for coming on the podcast.
Yeah, my pleasure. Thank you.
We hope you like listening to the podcast as much as we like making it. If you think it's valuable, share with somebody. Send to someone you think would really benefit, and leave us a 5-star review in iTunes. Those reviews really, really help us out. Find us on Facebook and follow us on Instagram @WildHealthMD. If you didn't like, well, why are you still listening? Email us and let us know what we can do better. But the best way you can thank us for our time is